Okay, this is the screencast on diffusion, osmosis, which are kinds of passive transport, and then active transport. This is going to be a pretty long screencast, so if you don't need to listen to the whole thing, you can fast forward at any time or jump ahead to another slide. Okay, before we talk about passive and active transport, how cells transport molecules within a cell, we need to know what the purpose is. And the purpose is to maintain homeostasis. Cells need a stable internal environment. In a cell, factors such as pH and the concentration of other substances must always remain the same, even if the external environment changes. It is the special properties of the cell membrane that help the cell maintain homeostasis. Again, an example of homeostasis would be human body temperature, 98.6 degrees Fahrenheit. We maintain that constant stable environment. Even if the temperature outside goes down or the temperature outside goes up, we still need to maintain that constant environment. That's called homeostasis. Now, this is some of the ways in which cells maintain homeostasis. Okay, we need a little background information, and this is a physics lesson that you probably had in junior high, but we'll just review it quickly. Atoms and molecules are always in motion. The atoms of gases are moving about the most freely and the fastest, but even the molecules of solids are moving about a little bit. They're sort of jiggling in place. That energy of motion is called kinetic energy. Cells use the motion of the molecules around them to come into the cell or to leave the cell. And that's the motion. The motion of energy is called kinetic energy. Now, diffusion is movement of molecules from an area where they're very concentrated to an area of lesser concentration. For example, if a student sprays perfume in one part of the room, eventually the students on the other side of the room can smell it. That's because of this kinetic energy. These molecules are always in motion and they move from areas of high concentration to low concentration. Eventually, we can no longer smell the perfume, but that doesn't mean it's not there anymore. It's just in such small quantities that our noses aren't sensitive enough. There's such a small concentration of the perfume molecule that our noses aren't sensitive enough. If you brought a dog in, maybe that's a good dog that smells, that has a good sniffer, not that smells good, whose nose is very sensitive, it might still be able to pick up the perfume even when we can't. Okay, a relevant question still having to do with this kinetic energy, this energy of motion. Do molecules diffuse faster or slower in warm temperatures? Are molecules moving around faster when it's warm, or are they moving around slower when it's warm? And the answer is that they move faster in warmer temperature. In fact, temperature is nothing more than a measure of how fast molecules are moving. You feel heat when the molecules, let's say when you go outside on a hot day, the molecules, the air molecules are hitting your face at a faster rate, and you'd interpret that as heat. On a cold day, the molecules are not hitting your face as fast, and you interpret that as cold. If you look at how a thermometer works, the molecules inside a thermometer, when they are moving fast, the alcohol inside of a thermometer, when those molecules are moving fast, they creep up the inside of the thermometer so that it looks like a temperature that we would say is a high temperature. But when the alcohol molecules in that thermometer are moving slowly, they don't creep up the tube as far, and we interpret that as a low temperature. Now, bringing it back to the cell, diffusion occurs across cell membranes, but primarily diffusion has to happen with molecules that are small enough to fit across the cell membrane. So it's determined mostly by the size, but also a little bit by the chemical charge. There are 4 molecules that you should know that can diffuse across the cell membrane. Oxygen, glucose, carbon dioxide, and water can diffuse across the cell membrane. So they don't need to be pushed by the cell, they just come and go. Now, the silly thing is, when we're talking about the diffusion of water across a membrane or across a cell membrane, it has a special name. It's called osmosis. So if sugar diffuses across the cell membrane, we just call that diffusion. But if water diffuses across the cell membrane, we have to call that osmosis. So it's just diffusion across a semi-permeable membrane from an area of high concentration to an area of low concentration. That's what diffusion is. Now on the last slide, I whizzed across the idea that water diffuses from an area of high concentration to an area of low concentration. What on earth does water concentration mean? Well, it's referring to how many water molecules there are in a given volume. I'm sure that you can imagine if you have a glass of water compared to a glass of syrup, you can imagine that the glass of water has more water molecules compared to the glass of syrup. There's water in the syrup, but just nowhere near as much. So the higher concentration of water would be the glass of water, the lower concentration of water would be the syrup. So which has greater water concentration, distilled water or salt water? Well, distilled water is 100% water molecules by definition. That's what distilled water is. So it has the highest water concentration. Salt water has salt dissolved in it, so salt water has a lower concentration. Okay, this slide has some problems, I'm afraid, with the animation, so we're just going to have to deal with it for a minute. Let me explain. When a cell is placed into a different solution of water, it causes the cell possibly to gain water or to lose water, and that therefore would cause the cell to shrink or to increase in size. And remember that cells want to maintain homeostasis. They want to always stay the same size. So there are three situations that three different solutions they can be placed in, and depending on the solution will affect the cell in different ways. The first kind of solution is called an isotonic solution. Iso means same. If you remember, an isosceles triangle has two sides that are the same. So an isotonic solution has an environment that has the same concentration of dissolved substances as inside the cell. So if you look outside the cell, it says 80% water and 20% dissolved substances. That's the water concentration, 80% water. The inside of the cell says 80% water. So what happens to this cell is water diffuses out at the same rate that water diffuses in, because the concentration is equal on the inside and the outside. So water comes and goes, but that doesn't ever affect the cell size because water is leaving and coming at the same rate. Now the next one is called a hypotonic solution. A hypotonic solution is when you have an environment that has a lower concentration of dissolved substances than inside the cell. An example of a hypotonic solution is distilled water. It's 100% water, and so it has no dissolved substances in it. And because it's 100% water, please ignore the other arrows, just the arrow that's closest to distilled water. Water rushes into the cell. It's going from an area of high concentration to an area of low concentration. So 100% water is more concentrated than 80% water inside the cell. So water starts to rush into a cell if it's been placed in a hypotonic solution, and that causes the cell to swell. Now a cell could be placed in another situation. It could be placed in a hypertonic solution. Hypertonic means that the environment has a higher concentration of dissolved substances than inside the cell. Look at the outside of the cell. It has 30% dissolved substances. Inside has only 20. What is important here, though, is the water concentration. There's only 70% water concentration on the outside and 80% inside. So inside, there's a lot of water pressure in comparison to the outside, and that causes the water to leave the cell. A cell placed in this environment is going to start shrinking. Water is going to leave it. Here's a good visual that's taken directly out of your textbook about this isotonic and hypertonic kind of solution. This is what would happen if you put a human blood cell in different solutions. Look at the bottom one. This is where you have an isotonic solution where the water concentration is the same on the outside of the cell as it is on the inside of the cell. That's ideal. That's what your blood is supposed to be. And notice that the blood cell has its typical sort of donut shape. That's the way that blood is supposed to be. The blood cell is supposed to look. Water is coming and going at an equal rate. If, however, we change the consistency of the blood and we make it so that it's a hypotonic solution where there's too much water on the outside and not enough water on the inside of the cell, the water starts to rush into the cell and it causes the cell to expand. Because this is an animal cell, that cell could eventually burst. If you place a human red blood cell into distilled water, it will eventually pop because it doesn't have a cell wall. It's got just that cell membrane. Vice versa, if you drop a human red blood cell into salt water or for some reason the blood itself becomes hypertonic, there's not as much water on the outside of the cell as there is on the inside of the cell. The concentration is lower. Maybe you put it in syrup, for example. The water is going to start rushing out of the cell, and it causes the cell to shrink up, and then the blood cell can't do its normal functioning. So what the cell is needing is for that isotonic solution. It needs water to be the same concentration on the outside as it is on the inside. Okay, now this is a photograph actually of plant cells. These are onion cells that you're going to see in the lab, in our Osmosis demo lab. These are onion cells. You've seen onion cells before. These onion cells have been placed in a red dye so that you can see the inside a little bit better. The cells on the left side were placed in distilled water, and the vacuole has filled completely up. The cell is absolutely bursting. It's got tons and tons of water pressure in it because that distilled water is 100% water, and it's rushing into these cells. They have an advantage though, because they have a cell wall. These cells can never pop, so they just gain high pressure, and that's actually healthy for the cell. Then immediately you can see the clock up at the top. They change the water solution that these cells are in, and they place it into salt water. The salt water has such a low concentration of water, 85% water, if it's 15% salt, the water starts to rush out of the central vacuole, and the inside of these cells shrivels up. The cell wall is still there, and it's still the same size, but the water inside that cell has shriveled up. So this is what it says. Hypertonic solutions cause the vacuole to shrink, so the plant wilts. It no longer has that high pressure that allows the plant to stand upright. Because of the cell wall, hypotonic solutions, the one on the left, the distilled water, cannot cause a plant cell to rupture. This leads to an increase in what they call turgor pressure. That's definitely a vocabulary word on the test. Turgor pressure, which is the pressure of the vacuole on the cell wall. And that's actually healthy for a plant. So the more pressure that the plant cells are under, the better able the plant is to stand upright. So putting a plant in salt water is absolutely detrimental to the plant, at least to a freshwater plant. Okay, this is another kind of diffusion that's a little bit trickier. It's called facilitated diffusion. On occasion there are, well, I'll read what it says here. Particular molecules can bind to special protein channels in the plasma membrane. Some molecules don't just diffuse through the cell membrane all by themselves, like water, oxygen, glucose, and carbon dioxide do. Some of them need a little bit of help to get through. So they use these protein channels that are embedded in the cell membrane. But the important part, the reason this is called facilitated, that means to facilitate something means to help it along. Diffusion means it doesn't require energy. So that little red ball, the red molecule, can move from the top of the membrane to the bottom of the membrane through that transport protein without using any energy. The energy is called ATP. We call it facilitated because it's helped by that protein, but diffusion because the protein didn't need to use any energy. It happens, it's like the molecule is traveling through a hallway, and the hallway doesn't need to exert any energy to allow something to pass through it. These proteins are kind of like hallways in the cell membrane. Now's where it starts to get a little bit trickier. We talked about passive transport, and I said that diffusion and osmosis are examples of transport that happen automatically, no energy is required. Same thing with facilitated diffusion, where the proteins in the membrane act like little hallways and the molecules just pass back and forth and no energy is required. That's called passive transport. But there's another kind of transport called active transport, and you can almost imagine by the wording how active transport is different. Passive transport is when materials move across the cell membranes without using cell energy. Diffusion, facilitated diffusion, and osmosis are examples. Water, oxygen, carbon dioxide, glucose, they can pass through using passive transport. They don't need any energy required to pass back and forth across the cell membrane. Active transport, which is the bottom diagram, active transport is when molecules have to be moved across the cell membrane and it requires the use of cell energy. There are two different circumstances where the cell actually has to push molecules across the cell membrane, and that's our next slide. The cell has to use active transport in two circumstances. The first time is when molecules that are just too big to diffuse across the cell membrane. We actually have to use transport molecules, transport proteins, and they use ATP to force that molecule across the cell membrane. So maybe if there's a nutrient or something that the cell needs, that it needs to push it across the cell membrane, or vice versa, if it needs to get something out of the cell, it needs to push it across that cell membrane. It takes energy to do that. There's also a circumstance where they have to normally the molecule would diffuse into the cell, or normally it would diffuse out of the cell. This is when the cell wants it to go the opposite direction. If the molecules are all leaving the cell and the cell needs those molecules, it's going to push them back in. So if normally they would diffuse out and the cell needs them in, it's going to push them in, and it's going to use ATP. It's going to use cell energy to keep those molecules back in, to push them, pump them back in. So this is when they has to move the opposite direction of diffusion. So again, 2 times that the cell needs to use active transport. The first time is to just push big molecules that can't just squeeze across. The second time is to make molecules go the opposite direction that they would normally go via diffusion. Okay, these are 2 big examples of active transport. They call this bulk passage into and out of the cell. Bulk means a lot of stuff is coming in and out. Endocytosis occurs when the cell needs something to come into it. So the cell membrane, do you see those little red particles in the diagram? The cell wants those red particles inside of it. So it creates this little cavity and creates, in essence, what is a vacuole, not a vacuole, a vesicle. There we go, that was the word I was looking for. The plasma membrane engulfs those little red particles, and that requires energy. There's also the opposite process, and you saw this with the Golgi. When the Golgi produces its, remember the Golgi is the stack of pancakes with blobs of syrup. Those blobs of syrup are called vesicles, and we need to push those vesicles out of the cell. So that's what you see in the diagram, and when it wants to expel anything from the cell, it uses active transport to push them out of the cell. So that's called exocytosis. It's the reverse of endo. Endo is in, exo exiting. Exo means getting out. So proteins or other molecules are secreted out of the cell this way. There are 2 things I will not test you on. They just happen to be on the screen. Phagocytosis is when the cell is bringing in solids or foods. Pinocytosis is when the cell is bringing in liquids, but I will not test you on that. Okay, this is another example of active transport. If you look at the diagram, you see ATP. This is a much smaller example. The last 2 examples are bulk examples, large examples of the cell expending energy to bring things in and out of the cell. This is a very, very tiny but super important example. It's called the sodium-potassium pump. I'm basically going to read it to you and then add a few little details. The sodium-potassium pump binds 3 sodium ions and a molecule of ATP. Look at the 3 orange balls. Those are sodium ions. The cell wants those sodium ions out of the cell, and it wants the potassium ions to come into the cell. They normally don't pass through the cell membrane via diffusion, so the cell has to work to get this to happen. So it binds those 3 orange balls, the sodiums, and it grabs a molecule of ATP. So second section, the splitting of ATP into ADP. Remember, ATP stands for adenosine triphosphate. Notice that it has 3 P's. Okay, when you take off one of the P's, it now becomes adenosine diphosphate. Notice it only has 2 P's now. That releases energy. So it says the splitting of ATP into ADP provides energy to change the shape of the channel. That purple protein changes shape. The sodium ions are driven through the channel and released on the other side. So slide 3, the sodium ions are released to the outside of the membrane, and the new shape of the channel allows 2 potassium ions to bind. Those green triangles are the potassium ions, and now they can bind, and they're on the outside of the cell. They're now going to come into the inside of the cell. In step number 4, the release of the phosphate allows the channel to revert to its original form, releasing the potassium ions on the inside of the membrane. So again, this is active transport. The potassiums are being pushed out of the cell. The sodiums are being pushed out of the cell. The potassiums are being pushed into the cell by using a molecule of ATP. We pop off that 3rd phosphate and it turns it into ADP, and that releases the energy necessary to change this molecule. All right, we're almost done. This is the last slide. The proton pump is the way that ATP molecules are made, where ATP actually comes from. So initially, this is what slide number 1 says. Energy from photosynthesis or food molecules when we eat is used to power a channel that transports protons across the membrane. So protons are H pluses, hydrogen pluses. That means that they've lost an electron and all they have is a proton, and so sometimes we refer to them as H plus, sometimes we refer to them as a hydrogen ion. In this case, they're referring to them as protons. So the protons are being moved across the cell membrane where they are collecting on the opposite side of the membrane, and that's what slide number 2 is. As the process proceeds, a high concentration of protons builds up on the far side of the membrane. So look at how many hydrogen protons are forming on that gold side of the membrane. Number 3, protons then diffuse back across the membrane. They're moving from an area of high concentration now to an area of low concentration. And these special channels use the force of the protons pushing through to power the manufacture of ATP. Where does ATP come from? It comes from ADP. If you remember in the last slide, ADP forms when we pop a phosphate off the end of ATP. Well, now this proton pump reattaches that third phosphate, so you are recycling, constantly recycling ATP and ADP. So this proton pump reattaches that phosphate, and now you have a new molecule of ATP ready to power other cell processes. That is it. Whew, that was a long one.